inflammation and implantation, which is the subject of my talk. And especially, my focus is going to discuss the aspect of what is the role of inflammation and how this evolutionary need for the success of the pregnancy. The work that I'm going to present is mainly work done by this fantastic group of investigators, members of my lab, initiated by Palome Aldo, Jay Yuan Kwan, Karen Rasicot, Yuan Yu. I'm going to show a lot of the animal models, Candy Ding, as well as Ingrid Cárdenas, and I will name another ones as we go through the presentation. So what is the problem? Really what we're trying to understand is more what are the aspects of implantation. And we know that implantation is the essential step for the success of the pregnancy, and is extremely important and highly demanding because it requires a nice coordination between the endometrium, the host, and the embryo, who is the visitor, if we want to call it, and they need to talk and coordinate in order to have the success of the pregnancy. For this group, I don't need to go to the details, you know very well the different stages of the pregnancy, but there is that critical stage that is called the window of the implantation. And this is the stage when the uterus has changed, as we will talk later, from a state that is an anti-implantation, because the uterus is covered by this mucin layer, preventing the embryo to implant, and there is a unique and a specific site in the uterus, and in a specific time, that is this window of implantation, where the mucin has been removed, there is additional molecules that allows the embryo to attach, and then have a successful implantation. 3 stages have been defined in general about the implantation stage: apposition, attachment, and invasion. However, we look at this a little more in detail because, as I mentioned, the surface epithelium of the uterus is covered by this layer of mucins who are preventing the attachment. So we need changes that I call the first phase of the attachment, that you need to remove the mucin layer, you need to induce the expression of additional molecules, and that leads to the first contact, that is the attachment of the embryo to the epithelium. But that's not enough for the implantation. The attachment represents only the first step. The completion of the implantation process will occur only when the tropoectoderm, cells from the tropoectoderm, the trophoblast, will start migrating from the embryo, going through the epithelial layer and invading the stroma. Once that is completed, implantation is successful. And implantation, it is a problem because, as you know very well, failure of the embryo to implant is clinically relevant to problems such as recurrent pregnancy losses. And the infertility, it is a problem. Is 15% of all the couples will have implantation failure, and is a social and economical problem. Therefore, trying to understand or improve the process of implantation will increase also the success of assisted reproduction, such as IVF. The receptivity or the capacity to receive the embryo by the uterus, by the endometrium, seems to be one of the limiting factors for the success of the pregnancy. We know multiple aspects has been described that may affect or lead to implantation failure. And we know that genetic or metabolic abnormalities, poor uterine receptivity, and there is this area that has been called the immunological rejection. My lab has been studying the immunological aspect for many, many years. And the focus of implantation failure from an immunological point of view, has been associated with the rejection of the maternal immune system into the implanting area. However, we know today that pregnancy, and especially at the time of implantation, is a unique immunological condition. Why? Because contrary to what we thought for many years, pregnancy is not an immune suppressed condition, number one. Pregnancy has a very strong and active immune system, I will discuss quickly. But also, at the implantation site, or during the early stages of the pregnancy, you have a lot of immune, almost all the immune cells that are present in the body, including T cells, NK cells, macrophages, dendritic cells, and so on. And many of these cells were thought to be associated with a rejection of the fetus by the mother. However, we know today that those immune cells present at the implantation site are not there because they are attacking the paternal antigens. They are there because they are required for the success of the pregnancy. Depletion of NK cells, for example, will lead to an incomplete trophoblast migration. Depletion of filioblattary cells will lead to a rejection, and so on. We talk in this lecture about the role of dendritic cells, and we will see later what they play during the implantation. But the first message that I want to leave with you is all the immune cells present at the implantation site are there to support the pregnancy and not to attack the pregnancy. The second aspect that I want to discuss with you is associated with the idea that pregnancy is an anti-inflammatory condition, which is true, but is not true. Pregnancy is a continuous evolving process from an inflammatory and immunological point of view. It's not in a static, and it's changing dramatically in every stage of the pregnancy. Initially, we describe pregnancy in what we call three immunological stages. The first trimester, we call it a pro-inflammatory stage. The second trimester is an anti-inflammatory stage, and the third trimester is again a pro-inflammatory stage. So what has been called pregnancy as an anti-inflammatory is true, but that occurs only during the second trimester and later trimester. Early pregnancy and late pregnancy are inflammatory, and I will discuss why the need of those conditions. So why do we need inflammation in the early pregnancy? And the reason is because implantation is an inflammatory process. What is the role of inflammation during this implantation period? And as I mentioned to you, where the inflammation plays a role. So let's review again. And that's what we're going to discuss in this lecture. What is the role of inflammation during apposition and attachment? What is the role of inflammation during migration and invasion? And is inflammation necessary for subsequent stages? That's what we're going to try to cover in the next few minutes. So what is the role of inflammation in the early stages of implantation? So first, inflammation and the preparation of the epithelium. Let me remind you again, the epithelium of the uterus is an anti-implantation organ because it is a mucosal surface epithelium. And that any mucosal surface epithelium is protected by a layer of mucin. That layer of mucin has two roles. One is prevent the migration of any type of microorganisms, of dangerous signals, such as bacteria and viruses, but also is housing in this mucin what we call today, that we know is the normal flora, the microbiome, the normal microbiome of the uterus. So this is always covered. In order to, as I mentioned to you, to have implantation, the surface of the epithelium needs to be changed. The mucin needs to be removed, and the adhesion molecules needs to be exposed so the embryo will attach. So the questions that we had when we initiated the studies are two mainly ones. One is, what are the factors that remove the mucin layer that is covering the epithelium? And second, what regulates the expression of the adhesion molecules? So in reality, the understanding of these changes came out from a study and a collaboration with Professor Nava Decker from the Weizmann Institute, because she reported that a biopsy of the endometrium induced an inflammatory process that may facilitate the preparation of the endometrium for implantation. Her report was associated with a clinical study where patients who have implantation failure, they will undergo a biopsy, and the original idea to do the biopsy was to take an endometrial sample in order to analyze the histological structure of the endometrium. The interesting thing is that they observed that many of those patients, not all of them, but many of those patients who had the biopsy, when they did the IVF in the next cycle, those patients got pregnant. So the question they had, and when we started the collaboration, is what that biopsy is doing to that endometrium that then facilitates the implantation of the embryo. So the study that we did consisted on evaluating those biopsies or the effect of those biopsies in the endometrium. So the study was taking a biopsy of the patient in the day 8 of the menstrual cycle, then taking a second biopsy in the day 21, and evaluating if the endometrium has changed. And we had controls where we didn't take the biopsy in the day 8. We just took one biopsy in the day 1, and that's what we compared. So we did first an evaluation of what immune cells who are present in the endometrium who had a biopsy or that did not have a prior biopsy. And we look at all the immune cells to see the changes. And the interesting thing is that we saw changes only in one cell type population of immune cells. And those were the HLA-DR positive dendritic cells. So with the graphic, what it shows you in day 8, when we took another biopsy in the same endometrium 14 days later or 15 days later, we saw that there was a significant increase of this group of cells, of these dendritic cells. Endometriums who have no prior biopsies, the number of dendritic cells did not change, even though it was in the day 21. So there was a major differences in the amount of dendritic cells between the two groups. The second aspect that we look at this endometrium is whether there were changes in the type of cytokines and chemokines expressed in the endometrium. So here, to facilitate what I'm showing you, the arrow indicates the groups who had the previous biopsy, but also, those are the patients who got pregnant. And interesting is to see that all the patients who got pregnant because they had a prior biopsy, they were characterized, the endometrium that we took was characterized by the expression of inflammatory cytokines, including GRO-alpha, IL-15, MIP-1 beta, and tumor necrosis factor, or TNF-alpha. So the picture that we're getting now is that in order to get an endometrium that would allow the implantation, you need to get dendritic cells and you need to get inflammatory cytokines. So we wanted to understand what is the role of each of these two factors. So the first thing is we wanted to know what is the role of dendritic cells. So for that, we moved to animal models. And in the animal models, what we did is to deplete the mouse of dendritic cells at the time of implantation. So what I'm showing you in this picture is an image of an embryo implanted in the mouse uterus, as you see here. And in the magnification, you can see the embryo embedded within the endometrium, within the decidua of the mouse. Now, into this mouse, we depleted dendritic cells starting in day, before implantation, in day E.1. And if we look what happened with the embryo, first of all, that by removing the dendritic cells, there was no pregnancy. So we look at the early stages, and you can see in this image that the embryo, which is looking here, is trying to attach to the surface epithelium of the uterus of the mouse, but is not able to do it. The epithelium is not receptive. As a consequence of the lack of this epithelial receptivity, the embryo will fail and the pregnancy will be terminated. So in the absence of dendritic cells, the epithelium of the uterus maintains its anti-receptivity, or receptive to the embryo. So as I mentioned to you, the non-receptivity of the epithelium is due to the presence of the mucin and the absence of addition molecules. So in order to test whether indeed the factors secreted by the dendritic cells are responsible of changing the epithelium, what we did is we took conditioned media, or again secreted factors from dendritic cells, and we exposed epithelial cells to those factors, and we look for the expression of mucin 16, CD44, alpha B beta 3 addition molecules, and so on. I show you for two because of time. Again, the epithelium, the surface epithelium, express levels of mucin 16, as demonstrated by the red color here, and it doesn't express CD44. However, if they are treated with conditioned media from dendritic cells, the mucin CD16 disappears from the culture. The expression of mucin 16 is inhibited, and now you have also a high expression of CD44 and addition molecules. So to summarize all these studies in one single slide, what we learned from this, and is published here in the references, is that dendritic cells secrete factors that will modify the epithelium of the uterus by removing the mucin and inducing the expression of addition molecules. The two primary stages that will allow the synapses between the trophoblast, the fetus, the embryo, sorry, the trophoblast and the embryo, and the surface epithelium in the mother. It's the first physical connection between the mother and the baby. So dendritic cells and the factors by dendritic cells are critical for preparing the attachment. The second question is, and I always have this question when I present to my colleagues, is this something that exists in nature? Is this, or is unique to humans, or is something that we observe just in the mammalian species? So in order to answer that question, we discussed this problem with Wagner Gunther, a professor at Yale, who is an expert in the evolution of reproduction. And the study that he did is he compared these three different mammalian species, but who have different type of pregnancies. All of them are mammals, the monotremes, the marsupials, and the eutherians. All of them will take care of the babies by producing milk. However, the monotremes, contrary to the other mammalians, like eggs. So they lay eggs, and what they have in common is that the female will nurse with, but again, this is an external pregnancy. The marsupials are getting closer to the humans, or to the eutherians, who have a placenta. The marsupials, they will have an implantation period. So the embryo will be inside of the uterus of the marsupials. However, they will carry the babies in the pouch. They will not carry the pregnancy in the uterus. And they don't have a placenta. All of them are mammals, I repeat. The eutherians, who are informally defined as placentals, what they have in common is that they develop a placenta, different type of placentas. And they will carry the pregnancy within the uterus until the end of the pregnancy. So here comes the interesting study by Dr. Gunther. He focused on the opossum, and the opossum belongs to the family of the marsupials. So they have a very short implantation period of 24 hours, and then the rest of the pregnancy will be carried in the pouch. So he analyzed how that is happening in the uterus of the opossum. And what he shows is, here is the embryo, who is inside of the uterus of the opossum. Here we can see, let me go back. Here we can see the surface epithelium of the uterus of the opossum, the stroma, the trophoblast of the embryo, and there is an attachment of the trophoblast to the surface epithelium. And as I said before, that attachment happens only during 24 hours. So it's a perfect attachment. Unfortunately, or what happened in marsupials, that attachment ends in those 24 hours, and there is a parturition. So there is a separation between the trophoblast and the surface epithelium of the uterus. The marsupial has a plan similar to pregnancy failure, similar to what I show you with the mouse that was lacking dendritic cells. The trophoectoderm tries to attach. It may succeed at the beginning, but then also will fail. The marsupial has a plan B, and that plan B is the pouch, and will carry the baby in the pouch. In humans and mammals, placenta, if there is no attachment, there is no plan B, the pregnancy will be terminated. But here comes the interesting aspect, because when he evaluates doing a race, RNA-seq, a race, what are the dominant factors produced at the time of implantation, as well as following implantation? This is what he found. The red arrow indicates the genes were induced at the time of implantation. And the star indicates the time following the implantation, that is the detachment. So the first thing, at the time of implantation, what he found is that the dominant genes were expressed are inflammatory genes. So similar to what I was showing you in the human as well as in the mouse, the process of implantation requires, the process of attachment. The process of attachment requires an inflammatory response. Inflammation is necessary for attachment. But here came the interesting aspect. The detachment was associated with the magnification of or the continuation of the inflammation. in terms of all the species. He evaluated multiple species like the sheep, the pig, horse, dog, mouse, and so on. In all of them, the process of attachment, the implantation, the early implantation, is associated with the production of pro-inflammatory cytokines. So, discussing with Dr. Gunter, I mean, how is that you have inflammation and then more inflammation and there is the detachment? So, if we associate his findings to the human, this is what we propose. That in the time of attachment, both in the opossum as well as in the human, you have an inflammatory process. As I mentioned to you before, and that is the difference with the opossum, we have a period that is a period of anti-inflammation. That is what allows the placentation and the maintenance of the baby or the fetus inside of the uterus. The end of the pregnancy, parturition, is associated with inflammation, as I mentioned to you before. So in the opossum, we see exactly that. Inflammation associated with the attachment, and a continuous inflammation is represent the time of parturition. So it's early implantation, early parturition. The opossum is a model of an implantation failure. So in humans, this transition from a pro-inflammatory environment into anti-inflammatory environment represents the evolutionary step that allows the maintenance of the pregnancy within the uterus, the growth of a placenta and the growth of the fetus within the organ, within the endometrium and the uterus of the mother. And at the end, similar as the opossum, you will have another inflammatory process which is responsible for the parturition. So we see now, in addition, what I mentioned to you, that this it's not that you have the inflammation, you shut it down and you open the anti-inflammatory and then you shut it down. It's a continuous process. It's a modifying process, and it's critical in terms of time and levels. And then this is what we show in a recent study that we published, where we found that when we look, for example, at two types of cytokines, IL-10 and TNF-alpha, that both of them are expressed in early pregnancy, starting at week 4 and going all the way to week 13. Let me just go and show you what I think is the most important. It's the ratio. It's the ratio between the two, the anti-inflammatory and the pro-inflammatory. So if we look at the ratio of anti versus pro-inflammatory, the early pregnancy, 4 or 5 weeks, has a lot of inflammation. As the pregnancy continues, we're talking about still the first trimester, the first 12 weeks, 13 weeks, around 6 to 8 weeks, that ratio starts changing. You have less and less inflammation. When you're reaching week 13, the ratio of anti-inflammatory is now higher than the inflammatory. And when that transition doesn't happen, that's when we see the pregnancy complications. Yeah? And here is this graphic can show you and explain better. The green is the normal pregnancy, the red is the implantation failure. And we're looking at the ratio of anti-pro-inflammatory. Again, early pregnancy in both, in the normal, those who will get a normal pregnancy versus those who have the miscarriages, early miscarriages, both of them has inflammation. So miscarriages are a good control for implantation, because you have a successful implantation. Again, it's a demonstration that miscarriages is a different disease than implantation failure. So both of them a full pregnancy or miscarriage has an anti-inflammatory that ensure the implantation. Here comes the difference. While in the controls or in the successful pregnancy, the anti-inflammatory start dominating and the inflammatory start going down. In the miscarriage, the inflammatory process remains high. There is no shift. And as a result of keeping in high inflammation, or you with the opossum, so we have an anti-inflammatory stage before implantation. In the early implantation, you have pro-inflammatory. If you want to maintain the pregnancy, you need to have this shift into anti-inflammatory. If the inflammation continues, then you will have a miscarriage. So that is in relation to the attachment. The next question is inflammation necessary for the parturition, for the migration and invasion of the trophoblast? And this I will show you very quickly because of time. So what we are trying to understand, again, this is a critical question, we still don't have the answer, is what are the factors who are making all these changes? Yeah. The signal who are maintaining this modulation going from one stage to another one is essential for the success of the pregnancy. Okay, so let's go to the implantation. One of the things that also we observe by using the biopsy that I mentioned to you at the beginning is that when there was a successful pregnancy, there was increase in the expression of TNF-alpha. So we wanted to know, what is TNF-alpha doing? Yeah. Is TNF-alpha acting on the embryo? And the answer is no. When we put TNF-alpha in trophoblast cells to see if the TNF-alpha that will enhance the migration and invasion, we couldn't find any effect. However, when we treat stroma cells, endometrial stroma cells, with TNF-alpha, we saw that the response of the stroma cells to TNF-alpha was the production of a lot of chemokines and growth factors, such as RANTES, growth alpha, MIB1 beta, GCSF, and GM-CSF. Those who are familiar with this area, you know that many of these cytokines, these chemokines, are essential for trophoblast migration and invasion. So, based on these findings, we hypothesized, and again, we know now that macrophages in the uterus in the early pregnancy are M1, and they are producing a lot of TNF-alpha. So going back, I say we hypothesized that macrophages are expressing or secreting TNF-alpha, and this TNF-alpha will act in the stroma cells, and as a result of this stimulation, the stroma cells will produce the chemokines that may lead or may help the trophoblast to migrate and to invade. So how do you see that? You know very well that we cannot do experiments with embryos. So what we did is we create an embryo-like structures where trophoblast cells that form these spheroids, and we will put them in a culture that has factors secreted by stroma cells treated with TNF-alpha. And what you see in the movie is that these trophoblast-like structures or embryo-like structures will attach to the plate, and the trophoblast cells will start coming out from this embryo-like or from this blastocyst-like structure and will cover the plate. If we do exactly the same experiment, but then we put, if we put a factor secreted from epithelial cells or from other cells, immune cells, the blastocyst-like structures of the trophoectoderm will not attach and will not migrate. So, and again, you see the time is exactly the same time they're still floating. They are in the presence of factors secreted by the epithelium. We did also put in T cells, B cells, and so on. The only factors that can induce what you see in the left side, attachment and migration, are factors produced by stroma cells stimulated with TNF-alpha. So to summarize these and more data that because of time I don't have, we know now that the macrophage is secreting TNF-alpha, and that is what is inducing the response. To show that indeed TNF-alpha is the one who is responsible of regulating those factors, what we did is knock down the receptor of TNF-alpha in the stroma cells. And then we collected the conditioned media, and we see it will be able still to induce the attachment and the migration. And the movie, what it shows you is that if we treat stroma cells with TNF-alpha, but those stroma cells do not express the receptor, now the capacity of attach and migrate is almost 90% inhibited. Again, confirming that the stroma cells require the receptor of TNF-alpha, and TNF-alpha responsible for the expression of the factors which will lead to the migration and invasion of the trophoblast. And if you remember the opossum will have an early attachment, but there will not be a migration. So what we did is we took conditioned media from the stroma cells of the opossum, we put it in this blastocyst-like structure, and what we saw, they tried to attach, when trophoblast cells are coming out, but they never succeed in forming a full migration and invasion. So that is the evolutionary difference between the opossum and a placentation, a mammalian placentation. The signals from the endometrium who are primed by TNF-alpha are capable of guiding the trophoblast to migrate and invade. So, and this is, we're showing you also the invasion using a three-dimensional model where we replicate the components of the uterus. I show you very quickly. This model has 3 layers, stroma cells in the bottom, matrigel or epithelial cells in the top, and then we put this spheroid that is made of trophoblast cells. If we put just in matrigel, nothing happens, there is no invasion, there is no migration, they don't move. If we put stroma cells stimulated with TNF-alpha in the bottom, you see a time-dependent migration of the trophoblast. Here's in the time. Today's is put it, it's attached, trophoblast cells start coming out, start invading within the matrigel, forming villa-like structures, as you can see, penetrating with that, creating a placenta-like in the petri dish. The interesting thing is that all these cells, all these trophoblast cells, are moving toward the stroma. The stroma is guiding these cells to form the structure. If we do exactly the same with stroma cells who do not express the TNF-alpha receptor, nothing will happen. They do not migrate. So what is a receptive endometrium? A receptive endometrium is the one who has an inflammatory process responsible of the epithelial modifications that I described to you. And second, as important as the first one, is an inflammatory process that is priming the decidual cells, the stroma cells. Because the stroma cells, we know now, are the ones that are responsible of guiding these trophoblast cells inside. When we do a wound with the biopsy, we are activating these stroma cells to produce TNF-alpha, and the TNF-alpha is acting back in the stroma cells to guide and make this endometrium receptive again. If that inflammation is not taking place, either because the dendritic cells are not produced, or because the macrophages are not producing TNF-alpha, or the decidual cells do not respond to TNF-alpha, the embryo will not implant and will not migrate. So to conclude, inflammation is necessary for the preparation of the epithelium for attachment. Inflammation mediated by TNF-alpha prepares the stroma to communicate with the blastocyst and to promote the process of migration and invasion. The lack of inflammatory signals is associated with poor attachment and or defective migration. So a successful pregnancy is the one which follows all these modifications in the inflammatory process. An inflammation at the wrong time or early inflammation leads to preterm birth. If there is not a shift towards an anti-inflammatory condition, you will have an attachment because you have inflammation, but you have a detachment because there is not a shift into an anti-inflammatory condition. And if there is no inflammation at all, there is an implantation failure. However, we also know that if there is excessive inflammation prior implantation, that is also detrimental, and there will not be implantation. So implantation failure may be due to a lack of inflammation or an excessive inflammation. And with that, and again, the message is, when we study these changes, you cannot base on one single sample, taking one single sample, one single measurement, because it's a modifying, it's an evolving process that is changing as the embryo grows and interacts with the maternal immune cells as well as the cells in the uterus. With that, I conclude, and I want to thank acknowledge the support from NIH, our collaborators, Dr. Nava Dekel, as well as many other people who have contributed to the study. And I thank you for your attention. And I hope that the distance will not be a barrier for maintaining a good communication. And again, thank you very much. And I wish a success in this meeting.
谢谢基姆教授给我们带来一个非常精彩的这个报告。炎症也是一直我们生殖免疫里面非常关注的一个问题，就炎症是好还是坏，到底这个是这个基姆教授给我们一个非常的一个清楚的一个描述，就是说炎症在这个妊娠过程中，它是存在一个不停的一个状态，正常的一些炎症的因子的一个表达变化然后是存在一个不断的一个动态的一个变化过程。那么这个炎症与种植的一个相关性，这个基姆教授也给我们非常的一个深刻的一个解释，特别是这个蜕膜里面的一些DC 或者 G 氏细胞，它是如何在妊娠过程中精确的一个调控这个炎症因子的一个表达，然后来提供给我们一个种植的一个最佳的一个窗口，然后如何去找到这样一个种植的窗口，也是我们非常关心的一个问题，并且这个炎症跟这个种植在不同的一个物种中也是非常的一个保守的，非常保守的，而且这个人为的去创造在内膜里面创造这个炎症，包括用TNF-alpha去处理，也能够提高这个内膜的一个容受性，然后增强这个胚胎的一个种植，这个确实对我们基姆教授的一个报告给我们非常的一个好的一个指示，也为我们以后的研究方向指明了一个很好的一个研究方向。我们再次感谢基姆教授的一个精彩的一个报告。